Hi, my name is Tiffany Aguirre, and this is the effects of chemotherapy on cognitive abilities utilizing the rat gambling task and novel object recognition task. So, while chemotherapy is extremely important in the battle of cancer, there have been reports of declines in cognitive abilities in patients who have either undergone or are currently undergoing chemotherapy, nearly 75%. This phenomenon is known as chemobrain or chemofog. Namely, attention, memory, learning, and decision making have been the subject of cognitive deficits being reported. The drug in this study looked at was paclitaxel, otherwise known as Taxol, an anti-neoplastic drug that is used to treat many different types of cancers. Another side effect is peripheral neuropathy, as paclitaxel is known to be a neurotoxin. While many patients report experiencing chemobrain during their chemotherapy treatment, suggesting an encoding issue in the context of memory, there have been reports of patients who have stopped chemotherapy and yet years later are still suffering from chemobrain, suggesting a retrieval issue potentially. In fact, after cessation of cancer treatment, up to 50% of breast cancer survivors have long-term cognitive impairments in verbal short-term memory, attention, concentration, information processing speed, or just executive functions in general. It is argued that the cancer itself may also be a factor of these cognitive impairments. So in order to tease that factor out, we are working with a rodent model that do not have tumors. So it is purely looking at the chemotherapeutic drug itself in order to potentially isolate the mechanism better. Use of the rat gambling task, a rodent version of the Iowa gambling task, where a rodent is trained to choose one of two levers. One lever being advantageous as it gives 2 pellets 90% of the time, and the other 10% will result in a 10-second timeout. And the other lever is disadvantageous as it gives 4 pellets 40% of the time, whereas the other 60% results in a 40-second timeout. This research is important because as the number of cancer survivors increase, the long-term effects of cancer treatment has become a significant issue. This project is aimed to look at whether the use of chemotherapeutic drugs such as Taxol will reduce cognitive abilities, and if so, whether injecting the rodent while the rodent learns a task, a potential encoding issue as mentioned before, would be different than injecting the rodent after it learns the task, which would be a potential retrieval issue. It was hypothesized that injecting paclitaxel into the rodent will cause long-term memory deficits and impairments in decision-making, and that injecting around the time they are learning the task will have a larger impact on cognitive deficits than injecting after. So we started out with 48 rodents, however the final number was 45. They were put on a food-restrictive diet until they reached 85% of their weight to increase food-motivated reward-seeking behavior. First, animals were subjected to the mechanical paw withdrawal threshold test to confirm there was no tactile allodynia prior to injection. Then they were trained to single lever press as well as dual lever press. After that, 2 groups were injected with 2 milligrams per kilogram of either Taxol or saline once every day for 3 days while learning the rat gambling task paradigm. The other 2 groups, while completing initial training, would immediately learn the rat gambling task paradigm, and then once learned, they would be injected with 2 milligrams per kilogram of either Taxol or saline. Once subjects completed injections, they were subjected to the mechanical paw withdrawal threshold to test for hypersensitivity. Then all animals were subjected to the rat gambling task paradigm for testing. Finally, all animals were subjected to the novel object recognition test to assess long-term memory. They were placed in a chamber with 2 identical objects and allowed to explore both objects for 10 minutes. Then, after 30 minutes, they were placed in the same box with one of the objects they had seen before, as well as a novel object. If they spent more time exploring the novel object, this indicates no memory impairment, as rats tend to innately explore new objects. However, if they spent more time or the same amount of time exploring the familiar object, that would be an indication of failed memory. Within our results, we did find hypersensitivity in animals injected with Taxol than in animals injected with saline, indicating peripheral neuropathy. However, there was no difference across groups of any decision-making impairments when subjected to the rat gambling task. In fact, all groups over time got significantly better with the task. There was also no difference across groups of any memory impairments when subjected to the novel object recognition task. These results may indicate that something else may be contributing to chemobrain, not just the chemotherapy itself. As suggested by others, the cancer itself may be contributing to the cognitive deficits. There have even been suggestions that old age may simply be the cause, as many patients who tend to report cognitive deficits are older patients with cancer. Or perhaps it could be all of these factors at play, only more research could tell. Thank you.